Welcome back to chapter 14, Blood. This is part 2 of the blood lecture. So last time in the previous video, I started off talking about, or I ended up, I should say, talking about the different types of white blood cells. And so as you can see, we have two main categories of white blood cells. You have your granulocytes and your agranulocytes. Granulocytes, those are cells that actually contain granules. And the agranulocytes are cells that do not contain granules. So the the granulocytes that we covered last video were your neutrophils, which turn light purple in with the acid-base staining. You'll notice that their nuclei have different numbers of lobes. In this case, you have 1, 2, 3, 4 in this teeny tiny little lobe right there. So that is a lobed nucleus. Other names for your neutrophils are segs, polymorphonuclear leukocytes, and bands. Bands are young neutrophils. This is the white blood cell or the leukocyte that makes up more than 50% of the total amount of white blood cells in the blood, 54 to 62%. And this particular white blood cell elevates or starts to multiply when you have bacterial infections. So when a person has a bacterial infection and the clinician does a CBC or CBC with DIF, they'll notice in that lab work that the person has elevated white blood cells, particularly neutrophils. Usually that means that the neutrophils will be greater than 62% of the total amount of leukocytes or leukocytes in that sample. Next, you have your eosinophils. Eosinophils turn a deep red primarily with your acid staining. So you'll notice that the cytoplasm still has lots of granules because this is a granulocyte, and those granules tend to turn red. And then you have your bilobed nucleus, bi meaning two. So you have these two portions, 1, 2, of the actual nucleus. This is the white blood cell that is elevated when you have either allergies or parasitic worm infections. So if a person, if you are as a clinician looking at the person's CBC or the CBC with DIF, and you notice that the eosinophils are increased, then you know that that person's problem, or at least part of that person's problem, is that they're having an allergic response at the time, or they're having a parasitic worm infection at the time. And as a clinician, then you can start to rule in and rule out certain things, asking the patient, what's been going on with them, have they ever traveled, etc., to sort of narrow down the possibility even more of either allergic reactions or parasitic infections. Eosinophils tend to be 1 to 3% of the total leukocyte composition. So anything above 3% eosinophils, you usually know that you have some allergic or parasitic infection on board. Basophils are the third and final of the granulocytes. You see all these grains, these little spots in the cell. These grains tend to be more purple because the base portion of the acid-base stain will end up turning them more blue. Basophils release histamine. Histamine you should be familiar with when you're talking about allergic reactions. Histamine is released in other times in the body, but this is where we know it the most because we tend to know of antihistamine medications that are decreasing your histamine response. It also releases heparin. Those of you who are in pharmacology now should recognize heparin. If not now, soon you should be able to recognize heparin as a blood thinner, the medication used as a blood thinner. This is a chemical that your white blood cells, specifically your basophils, produce to do the same thing. Basophils should be less than 1% of your leukocyte total. So if you have 1% or more leukocytes are basophils, then you know that something is going on with the basophils. Basophils are also similar in size to your eosinophils. Now let's look at your agranulocytes. Look at the picture, you'll notice that there are no granules, no spots. That's just why it is an agranulocyte. The nucleus tends to be more spherical or kidney shaped. So you see here, it looks more like a bean, a kidney bean, or kidney, I should say. This is a large white blood cell. It's the largest of all the white blood cells, and it's definitely larger than your red blood cell. These will leave the bloodstream as necessary to become macrophages. So macro means big, phage refers to eating. So this is a big eating cell. So when it leaves the bloodstream, it's leaving because there's something there, either bacteria or other dead cells. It could be your regular body cells that are just past their prime and have died, or some other debris, like if you get stuck with a what's the word? A splinter. That's what I was trying to say. You get stuck with a splinter, then the splinter is going to leave other kind of debris in there, little wood pieces or whatever else is on there. Your macrophages, so these are monocytes that leave the bloodstream. Once they leave, become macrophages. These big eating cells will then roam around the area and look for bacteria, dead cells, or other debris to start to gobble those up and dispose of it so that the rest of your body can go in and start to clean and repair the area. Lymphocytes are the second and last of the agranulocytes. You'll notice no granules. This is slightly larger than a red blood cell, so this one is going to be the one that's approximately the same size as a red blood cell, a little bit bigger, but this is going to be closer to the size of the red blood cell than any of the others. It has a large spherical nucleus surrounded by a thin rim of cytoplasm. So you see this one big nucleus in the middle, surrounded by the thin rim of cytoplasm around the nucleus. That is the telltale sign of a lymphocyte. Lymphocytes are T cells and B cells. When you hear conversation about T cells and when you hear conversation about B cells, you're talking specifically about lymphocytes, one of the 5 types of white blood cells. Both of these are important in immunity. So your B cells are the ones that are responsible for producing the antibodies. Antibodies, remember when we talked about blood typing in lab, we talked about antibodies. Antibodies are what your body produces against various antigens that are presented by bacteria, viruses, cancer cells, and foreign material. So these B cells will produce the specific antibodies that are against those cells. Of those leukocytes, lymphocytes will make 25 to 33% of them. So it's going to be about 1/4 to 1/3 of the total leukocyte population. If you have less than 25%, then you have a problem with leukocytes, or there's something else going on within the body so that something else is much higher percentage than it should be. And if you have more than 33%, then you may also have an issue with lymphocytes. So the function of your white blood cells. White blood cells protect the body against infection. And as you can see, each white blood cell has a particular job to do so that it is protecting against whatever type of infection may be occurring. So this might be your bacterial infection, your viral infection, your parasitic infection. It could be allergy. So anything that is there to attack the body that is not supposed to be in there, your white blood cells are there to protect you against those things. Leukocytes, some leukocytes can leave the blood vessels and go into the surrounding tissues. That is called diapedesis. It leaves the capillaries through the spaces between cells. So this is one cell, this is one cell, this is one cell. Each of these cells connect together to form the walls of the blood vessels. And in between these walls, kind of like sutures, not sutures, but kind of like sutures, these white blood cells can squeeze their way through these openings and create, squeeze their way through the openings in order to leave the capillary to go into the interstitial space and look for whatever invader it needs to look for to help clean up the area, disinfect the area, and get the area ready for the rest of the body to be able to go in and heal that area. So make sure you're looking at this. Make sure you are looking at, looking at this PowerPoint so that you can get a better understanding. If a person gets stuck with a splinter, then this is what this is blown up. The green bits are your bacteria that have been introduced into the body. You have your white blood cells, your neutrophils. So remember, that is the most abundant and the first white blood cell that's going to go to an area to help to clean up the area. These are arteries, these are veins that have been broken because of the action or the introduction of the splinter. So these blood vessels broken, blood seeping into the area. The neutrophils have been signaled. Since they are already the most abundant, they're going to be the first ones there. You have white blood cells and platelets also in the area. The platelets are going to help to eventually close off this damage so that you're not bleeding out. White blood cells are going to continue to gobble up these bacterial cells. You have the injured cells here that will release histamine. This is going to, remember I told you histamine is not just for allergy responses, but it also has other function in the body. So in this particular case, these cells are going to release histamine so that you can dilate or open up the blood vessels to allow more blood and more of those healing factors to go into the area to clean up and heal that particular area. This talks about the neutrophils moving through the blood vessels, and then you also have the neutrophils that are cleaning up and gobbling up the bacteria. So pay attention to this, look at all the parts, refresh yourself on different tissue types, your epidermis, your dermis, the different layers of your epidermis. Can you tell me what structure this is, or what one of two structures this might be? Just by looking at this, you should be able to tell me that by now. Yeah, so make sure you're looking at this very carefully so that you can understand what is happening. So blood cell counts. If you've ever heard of someone ordering a CBC or a CBC with diff, now you'll understand what those mean. A CBC is a complete blood count. Literally, they are counting how many blood cells are in your bloodstream. The way they do that is they take a sample of your blood. It can be a small tube of blood. It can be a drop of blood, depending on what all they need blood from. But they take a sample of blood. Then they drop a drop of blood onto a special microscope slide. microscope slide is going to have these square boxes, these really, really tiny square boxes etched into the glass. So that when you look at this microscope slide, under the microscope, you'll be able to see those little boxes. And once you put a drop of blood in there and you smear it, so you did kind of thin the blood out, you can see individual blood cells, you'll be able to count them. And so, excuse me, you'll notice here that it says 5,000 to 10,000 cells. This is what it's talking about, cells per cubic millimeter of blood. You're able to count how many blood cells you have per cubic millimeter per little box, and then you can multiply that by the total volume of blood. If your white blood cell count is below 5,000, so below this lower end of normal, it is called leukopenia. Remember, leuko means white. Penia, we talked about that when we talked about osteopenia. So it is a deficiency or a low amount of. So in this case, leukopenia is a low amount of white blood cells. Some causes of leukopenia can be typhoid fever, flu, measles, mumps, chickenpox, or AIDS. So when we are doing these diagnoses, we want to diagnose the thing, so we can diagnose leukopenia by just looking at the white blood cell count. But in order to treat the leukopenia, we need to know specifically what is going on with the person's body. And we have to rule in or rule out certain things. And this is why doctors and nurses do so many different tests. Leukocytosis. Leuko again means white, cyto means cell, osis is a condition of. So this is a condition of white blood cells. This is when you have high white blood cell count, so above 10,000, which is the upper end of normal. Leukocytosis can be caused by acute infection. So if you are ill and you've been ill just for a short amount of time, your white blood cells are going to increase quite a bit to help your body to heal from that infection. It can be vigorous exercise. Think about it this way. When you are exercising, especially exercising vigorously, you are eventually going to start doing anaerobic exercise, meaning you're not going to be able to consume enough oxygen to keep up with the demand, the oxygen demand of your cells. So your body will have to go into different forms of producing ATP. If you continue to work out vigorously, your body will adapt. Remember, your body is extremely adaptable. So the more you do vigorous exercises, the more your white blood cells will increase in order to help your body continue doing the work that it needs to do. You'll also have loss of body fluid. So when you lose body fluids, that is another signal for your white blood cell count to increase above the normal range. So your CBC is a complete blood count. What they'll usually do is run it through a machine. Excuse me. Excuse me. That machine will count the different the amount of red blood cells and the overall amount of white blood cells. Excuse me. When you do a CBC with differential or a CBC with diff, then it's going to go more specifically looking at how many of each type of white blood cell. So CBC with diff or differential will look for the 5 different types of white blood cells and count each of them so that it can give you a percentage of those specific to that, whatever is going on with the body. If you are healthy, then the percentages should be what was listed previously. If you have some disease process happening, then the number of or percentage of those particular white blood cells will change. So a recap of what can cause an abnormal number of white blood cells. These are all talking about elevated white blood cells, except this last one. This is what can happen if you have the illness that is caused by having too few, specifically lymphocytes or helper T cells. So use this to help you get an understanding of those. Ignore that. Blood platelets. So blood platelets are actually called, they're also called thrombocytes in the literature, in your textbook. Instead of seeing platelets, you may see thrombocytes. It's the same thing, it's just a different term for it. Thrombocytes are not actually cells. They're actually pieces and parts of cells called megakaryocytes. So the cells that the platelets come from, or that the platelets are fragments of, are called megakaryocytes. Platelets lack a nucleus. Again, they are pieces of a cell, so it won't necessarily have a nucleus because it's not a full cell. And these parts, these pieces, are roughly about half the size of a red blood cell. So pretty small. There are approximately 130,000 to 360,000 platelets per cubic millimeter of blood. Remember, these are cell fragments. The blood platelets, or the thrombocytes, help to repair damage in the blood vessel by sticking to broken surfaces. So if we go back to that image that we saw here, we'll see that you have a red blood cell that has been damaged. You have a white blood cell. I'm sorry, this is a, not red blood cell. This is a vein that's been damaged. This is a vein that's been damaged. The blue one is the vein. The red one is the artery. In between these small ones, this is capillaries. And up here, all of those things have been damaged. Also, this connective tissue has been damaged. This layer of the stratum basale, the other layers of the epidermis, all of this tissue has been damaged. So the platelets will go in, stick to the surface of the veins, I'm sorry, the arteries, the veins, the arterioles, the venules, and the capillaries, to start to close off the bleeding and to decrease the amount of damage, or close off the bleeding so that the body can go in and heal the damage. Let's go back. All right, so like I said, they help repair damaged blood vessels by sticking to the broken surfaces. Blood plasma. So blood plasma is the liquid portion of blood. The plasma itself is straw colored. So those first couple slides of this chapter, you notice that it was like this light yellow color. That's because that was the plasma, and it's normally that straw color. Of the total blood volume, that is the blood that is drawn directly out of your arm, 55% of that is going to be blood plasma. I know that the whole thing looks red, but that's because the red blood cells are so abundant. But once you spin it down, or once you allow the solid bits to settle, then you'll notice that the volume of blood, a little bit more than half of it is plasma. Of that plasma volume, of that plasma volume, 92% of this volume is water. So only 8% of this is going to be other things, including nutrients, gases, vitamins, waste products. And it helps, the blood plasma helps to regulate the fluid and electrolyte balance, and maintain your body's pH. All right, so these nutrients, these vitamins, these gases, all of those things will help regulate fluid and electrolyte imbalance, and they'll also help to maintain your body's pH. And each part of your body has a specific pH that it's dealing with. Your stomach obviously has a completely different pH than your big toe, which will have a different pH than your intestines. So every part of your body will have a different pH, and your blood plasma will help maintain the pH in those particular areas. Plasma proteins. So plasma proteins make up a good bit of the dissolved product in your plasma. Of the plasma protein, you have three main kinds of plasma protein. You have albumin, globulin, and fibrinogen. Albumin is going to be the most abundant of the proteins that are dissolved in your plasma. It is going to be about 60% of the total plasma protein volume. Albumin is made in the liver. And one of the things that it does is it helps to maintain that colloid osmotic pressure. So it helps maintain the pressure inside the blood vessels. Globulin, these are alpha and beta globulin are produced in the liver. Gamma globulins are produced in your lymphatic tissues. And these will help to do different things. Some will help to transport lipids and fat-soluble things throughout the blood. Remember that your blood is made of mostly water. And these are fat and fat-soluble products. And oil and water, fat and water don't mix. So in order for these lipids and these fat-soluble proteins to get from one place to another in the body, they have to attach to proteins. Some of them will attach to alpha globulins, some will attach to beta globulins, and many will actually attach to albumin in the blood to be transported from one place to another. Gamma globulins are important in your immunity. This is what is making up your antibodies. And this is, again, made in the lymphatic tissue. And then fibrinogen, which is has the smallest amount of plasma proteins in the blood. This is also produced by the liver. This has a key role in blood coagulation. So when we're talking about blood clotting or blood coagulation, it is a series of events. It's not just one thing in the body that does it. It's not just platelets. It is a series of events. And fibrinogen has a huge role in that series of events to cause your blood to coagulate or to clot. So do know the different proteins in the blood, the percentages of them, where their origin is. Most of them are liver, except the gamma globulin, and something about their function. So now, when we talk about gases and nutrients, the most important blood gases are going to be oxygen and carbon dioxide. There are other gases in our blood, but we mostly focus on oxygen and carbon dioxide. The plasma nutrients, again, this is not an exhaustive list, but you have amino acids. Amino acids are the building blocks of protein. You have simple sugars. Simple sugars are the building blocks of carbohydrates, as well as glycogen. Nucleotides are the building blocks of your genetic material, and lipid is the building block of fats and cholesterol. So all of these things are floating around in the plasma portion of your blood in order to transport it from one place to another so your body can have those items that it needs to survive. So then we're talking about the non-protein nitrogenous substances. Non-protein nitrogenous substances. These are molecules that contain nitrogen but are not proteins. A lot of the nitrogen that is produced in your body is used in proteins, but these substances are not proteins, though they have nitrogen in them, and they include urea, uric acid, amino acids, creatine, which are two different things, and BUN, blood urea nitrogen. So urea is a product of protein catabolism. Remember that metabolism have two different forms. You have anabolism, and catabolism. So metabolism is the overall term, which has two different portions or two different actions, catabolism and anabolism. Catabolism is breaking things down. Anabolism is building things up. So urea, product of protein catabolism, meaning that you get urea when you catabolize or break down protein. About 50% of the non-protein nitrogenous substances in the blood comes from this urea, which is a product of protein breakdown. All right. Then you also have uric acid. This is a product of nucleic acid catabolism. So it's really important for you to understand the difference between catabolism and anabolism, because being able to understand these particular things is going to be hinging upon you understanding these. So again, uric acid, product of nucleic acid catabolism. That means you need to know where nucleic acid comes from. Amino acids. Amino acids are the product of protein catabolism. As I mentioned, amino acids are your building blocks for protein because protein had been broken down to give you the individual amino acids, and then you can take those amino acids and you can put them all together to form new proteins. Creatinine is used as a phosphate store. So if you remember when we talked about the muscles last semester, creatine is a substance that your body makes, but it is also a product that people who tend to go to the gym a lot, they take this as one of their supplements, because creatine will help to restore ATP molecules, because creatine stores the phosphate. Phosphate is part of ATP. ATP, adenosine triphosphate. So when you have an ATP go to an ADP, that's try to die, you've lost a phosphate group. And so when you want to bring back the ATP stores, you can use creatine to act one of the phosphates in creatine to restore that ATP from ADP. Then you have creatinine. Creatinine is the product of creatine metabolism. So as our bodies go through creatine, then the product that it produces is creatinine, because it now has less phosphates, because that phosphate was used to restore ATP. And then you have blood urea nitrogen. So this will indicate the health of your kidneys. How high or low this number is a direct reflection on the health and the functioning of your kidneys. So plasma electrolytes. Blood plasma contains a variety of ions. These are charged particles that are called electrolytes. So if you remember, your magnesium has a negative charge. Your calcium has a positive charge. Like, all of these things have either positive or negative charges. And because they are charged particles, they're called electrolytes. These electrolytes are absorbed from the intestines, meaning whatever you eat contains electrolytes, and those can be absorbed from the digestive system into the body, or they may be released as a byproduct of cellular metabolism. So when the cells metabolize, then they may release electrolytes as a part of their byproduct. These electrolytes include sodium. Usually you'll see sodium paired with chloride, but that's not the only thing it pairs with, but that is what it's mostly paired with. Sodium chloride, as you know, is table sugar. Potassium is another electrolyte. We'll often see potassium chloride, potassium bicarbonate, potassium sulfate. So there are different types of potassium salts. Calcium, magnesium, chloride again, bicarbonate, phosphate, and sulfate. All of these are electrolytes. Sometimes you'll see them as a salt. So again, potassium sulfate, magnesium phosphate, sodium bicarbonate, potassium chloride. All of those are salts, and each salt is made of these individual ions. And that is the end of chapter, what chapter is this? Chapter 14, your blood chapter.